Hola, las chicas! Okay, there are going to be two components to today's screencast. The first part is going to be about the cell membrane. And the second part is going to be about the cell theory. So, two basic components. I'm going to center this better. Cell membrane and cell theory. So let's get straight to it. Okay, cell membranes. What are they composed of? That's right. You got it dead on. They're composed of phospholipids. How did you know that? Okay, so a phospholipid has two basic components, or actually three components. The first part is fatty acid tails. It has two fatty acid tails. One is saturated. It means it's a straight line, right? Be solid at a room temperature. And the other is unsaturated, so it gives it this kink in the molecule, and it's a liquid at room temperature. So it's really important, actually, that a phospholipid has both an unkinked and a kinked fatty acid tail. It gives it a fluidity in the membrane, which you're going to hear more about a little bit later in the screencast, but it's important to know now that this fatty acid tail lends a fluidity to the membrane. Okay, and then it's got a glycerol head right here, and a phosphate group, which is very, very polar. So we've got a polar section to the molecule, which means it's hydrophilic, it interacts well with water, and we've got a very non-polar section of the molecule, which means it's hydrophobic, it hates water. So if you take a phospholipid, actually I'm just going to draw a schematic of a phospholipid right here. Let's see, I'm going to move it over. So here's a single little phospholipid with our kinked tail, and move it over here. Okay, phospholipid dude. I'm going to take a bunch of these and replicate them for you. Okay, so if we take a bunch of these phospholipids and line them all up next to each other, this is how they're going to align. All the hydrophobic tails are going to be next to one another, and all the hydrophilic heads are going to be next to one another. And if we put them in a water solution, H2O, over here, then the hydrophilic heads are going to be very happy, but as you can imagine, the hydrophobic tails are unhappy. But what if we make a bilayer? What if we take this, these phospholipids, and go like so. Now we've got two layers of phospholipids, and both sides are happy because we've got over here our hydrophilic heads interacting with water. Ooh, that's just an H2 there. I need an O. H2O. So our hydrophilic heads are interacting with the water, and our hydrophobic tails are interacting with one another. But, oh god, I forgot H2O over here too. You can imagine if we grabbed this water and put it on this side, now this is no longer happy. So, what if we extended these tails further out? Okay, so here you can see I've extended the membrane a little further, and we can go even further than this. If we take this whole thing and flip it, rotate it this way, you can start to see we're getting something that looks remarkably like a cell membrane. Indeed, because it is a cell membrane. So here we go. Zoom way in. We've got, very simple, just a single phospholipid interacting with water on one side and hydrophobic tails interacting with themselves on another side. And then it's actually a bilayer. There are two layers. So phospholipids on one side interacting with phospholipids on the other side. There's an aqueous solution, both interior and exterior. And we've created this perfect little cell. So none of the tails are interacting with water. All of the heads are interacting with water. Everyone's happy in this molecule. So this is basically a ball of fat. And that's what a cell is. It's just a ball of fat. And why do you want to be a cell? Why do you want to be inside a ball of fat? Well, it's warm, it's safe in here. If you imagine the exterior environment of a cell, it's very dangerous and scary. Lots of things trying to kill you. Here's a dagger trying to stab you. Lots of chemical explosions. Being enclosed in a nice little happy fat bubble, you are warm and safe and happy and protected from the outside environment. And that's really all a cell membrane is. It's a lipid bilayer, a ball of fat. But there's actually more to just fat that's in the membrane. I'm going to introduce you to a concept. It's called the fluid mosaic model. And I'm going to embed a picture here for you. Okay, so here's our picture, and if we zoom way in here, you can see that we've just got our phospholipids. They're the polar heads, and then the tails sticking at each other, and we've got our bilayer, 1 layer, 2 layers, bilayer. And the reason why this is called a fluid mosaic model for the cell membrane is that this membrane is not fixed. So this little dude right here, this phospholipid, right now he is in the left-hand corner of our membrane, but there's actually free diffusion happening within this membrane. So if we tag this guy with, say, a phosphorescent tag, and then we leave it for 5 minutes and we come back, and we might actually find that he has diffused all the way over here. So there's free movement of phospholipids within the membrane. And additionally, everything that's in the membrane, that's embedded within the membrane, is moving around. That's why we call it fluid mosaic model. Okay, so that's where the fluidity comes from. And the mosaic just refers to the fact that there aren't just phospholipids in here. You'll notice that there are cholesterol. Cholesterol helps regulate the fluidity of the membrane. There are proteins shown both across the membrane and also embedded on top or within the membrane. And there are also sugars, carbohydrates, embedded within the membrane. So it's a fluid mosaic model because things are moving around, and it's a mosaic. It's a bunch of different tile pieces all put together that make our cell membrane complex. Okay, that's it for the cell membrane portion of the screencast. Moving on to the cell theory portion. Okay, so the cell theory is pretty simple. You all probably understand it already, but it's worth reviewing right here. Okay, so the cell theory is composed of 2 components. The first one is that all living things are made up of cells. This is something you already know and understand when we talked about the different requirements for life. This is one of the things that all living things share, this common trait. They're all made up of cells. And the second piece of the cell theory is that all cells come from other cells. Which I know begs the question, where did the first cell come from? And we'll be talking more about that later on in the year, but it's a big question, and this is going to be a short screencast, so there's no time. So, punting that to later on in the year. So cell theory, all living things are made up of cells. All cells come from other cells. And there are a bunch of different types of cells. Cells come in 3 broad classes. The first is unicellular. Unicellular. The second is colonial. And the third is multicellular. Now, examples of unicellular organisms include bacteria, some fungi, and protists. Now, bacteria fall into one type of classification of life, and that's prokaryotes. Prokaryotes, as you recall, lack a nucleus and lack any membrane-bound organelles. Fungi and protists, however, belong to a different classification or a different grouping, and that's eukaryotes. Remember, these have nuclei and true membrane-bound organelles. Prokaryotes in general are much more simple than eukaryotes, but please don't confuse simplicity and complexity with success in life. There are actually many, 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 many more prokaryotes on Earth than there are eukaryotes all combined. Okay. Multicellular organisms include animals and plants and some fungi. And these are all examples of eukaryotes. Again. And then colonial organisms include some groups of protists. I'm going to give you a few different examples here. I'm going to embed a few photos and then talk about each of them. Okay, so here's our first photo. This is a micrograph of a bacteria. You'll notice it has a cell wall and a nucleoid, which is a nuclear region where the DNA lies, but again, no true nucleus. It also has ribosomes, which are important. You guys know what ribosomes are from filling out that organelle chart. So very simple structure, all single-celled, all unicellular. Other examples of single-celled eukaryotes include yeast, which are a single-celled fungus. You can see some membrane-bound organelles there. And also here's another kind of cool critter. This is Euglena. Euglena is very interesting because, as you'll notice, it's green. These green sacs are actually chloroplasts. So this dude is photosynthesizing, but he also has a mouth region and can consume things. He's also eats things. So he's what's called a mixotroph, performing both photosynthesis and also eating other things. And they've got little tails that allow them, that are called flagella, that allow them to swim around. We'll be looking at them underneath the microscope in a little while from now. But again, very cool organisms. Okay, I'm not going to embed pictures of animal, plant, or fungi cells besides this yeast, but I am going to show you an example of a colonial organism. This dude right here is an example of a colonial organism. It's a protist. It's also green, so surprise, surprise, filled with chlorophyll. It's got chloroplasts. It's producing, or it's performing photosynthesis. And so each of these dudes, like this big round ball, is an example of a volvax. That's what this is called. And a volvax is made up of a colony of a bunch of little cells. So each little green dot that you see here, so like that green dot is a single cell. This green dot is a single cell. This green dot is a single cell. And all their membranes are touching one another, and they form these spheres. They're actually amazingly beautiful to look at underneath the microscope. And each cell has a little flagella, one of those little tails, and they're phototactic. So if you shine a light on one side of the microscope, the whole ball will swim over towards the light because it's trying to perform photosynthesis. And actually enclosed within each globe or each sphere are these teeny little spheres as well, and these are actually the volvax babies. So when the time comes, this larger sphere will split open and all the babies will be released into the outside environment. So you might be wondering, well, what makes a colonial organism different from a multicellular organism? And the answer to that is that each of these cells that make up the volvax are identical in structure to every other cell that makes up the volvax, as opposed to an animal or a plant, a multicellular fungus, which have many, many different structures. So, for example, in us, we are multicellular organisms, and our heart tissue is very different from our brain tissue, which is very different from our skin tissue, which is very different from our blood tissue. So, in colonial organisms, all the cell types are the same, and even though they're living in a group, they're still not multicellular, which requires many different cell types living together. Okay, that is it for today. I will see you all tomorrow.